Hello everyone. The picture on this slide is that of a young lady who actually is experiencing a particular situation, a health condition caused by lupus. Lupus is basically an autoimmune disease, a disease by which your own immune system is attacking your own cells, your own tissues, which is a problem. So that takes us to the lymphatic and the immune system. Now, as we look at the lymphatic and the immune system, I want you to consider a number of things. One, it's what are the different parts of the lymphatic system and what are the functions of the lymphatic system and the immune system. What are the first and second line of defense in an innate immunity or non-specific immunity? In terms of specific immunity, what is cell-mediated immunity? And what is antibody-mediated immunity? What is active immunity, passive immunity? And what are allergic reactions? What is the basis of tissue rejection? And what are some immune system disorders? These are questions that we are going to try to address in this chapter. So the first step, it's going to be addressing the functions of the lymphatic system. The lymphatic system, it's a network of of lymphatic vessels and lymphatic capillaries. The lymphatic capillaries are going to absorb the excess fluid from the tissues. And as they take up this excess fluid from the tissue, they return that excess fluid in the bloodstream. And based on the volume of fluid in the blood, that excess fluid in the blood now can come back to the lymphatic system. The lymphatic capillaries carry a specific name. They are called lacteals. Lacteals in a particular area of our body in the small intestine, they are going to be essential for the absorption of fats. These fats are going to be typically associated with proteins, what we call HDL and LDL, high density lipoproteins and low density lipoproteins. But let me close that parenthesis. The lymphatic system is going to work alongside with the circulatory system. And their job together is going to be very important for the production, maintenance, and distribution of protective cells in the body, the white blood cells, right? And the white blood cells, leukocyte, are coming in different kind. We are going to address this different kind as we go through the immune system. And the previous chapter also gave us the chance to address the different kinds of white blood cells. So please review that chapter. The lymphatic system actually helps in the defense against pathogens, but also against cancer situations, right? Because cancer cells, even though they are our own cells, but these cells are behaving abnormally, they have to be destroyed and get rid of. Now, what are the different components of the lymphatic system? Well, we are going to have the organs that make up the lymphatic system. Some of the organs are going to be the lymph nodes, the lymphatic vessels, but we also going to have, as part of the lymphatic system, the lymph itself, the fluid, right? So that fluid is part of our defense system. So now on the left hand side, let's go step by step. We have the right lymphatic ducts. The right lymphatic duct is going to empty the lymph into the subclavian vein right here. And the clavicle is right here. The subclavian vein is going underneath the clavicle, right? So this is where the lymph is going to be emptied from the right lymphatic duct. Other organ we want to mention, the axillary lymph nodes in the armpit. The axillary lymph nodes are going to store leukocytes. The thoracic duct, so right here, the thoracic duct, it's going to receive the lymph that comes actually from the subclavian vein, right? So it goes like this, down, going down. Then we have the inguinal lymph nodes. These are the lymph nodes that are found in the groin areas. We're going to have the tonsils that are found in the nasobuccal area. They are going to be patches of lymphatic tissues. They help prevent entry of pathogens below the mouth, right? So in the mouth and the nose, the tonsils are going to play a role of protection so that the pathogens do not go further down. We have the red bone marrow. This is where all the blood cells are going to originate. In the case of the lymphatic system, we are going to focus on the white blood cells. The thymus, which is an organ that sits right on top of the heart, it's an organ which job is going to be to teach our protective cells to learn to set the difference between self and non-self. They're going to learn to tell apart what is ours and what is not ours. And finally, the spleen. The spleen is going to be very important at cleaning the blood, to get rid of debris, including dead bacteria. We are also going to have there resident lymphocytes, okay, white blood cells that are going to reside there and ready to respond to the presence of antigens or pathogens. So when we look, we zoom in the tissues. So from here, if we zoom in, what we see, we are going to see the lymphatic capillaries right here. These lymphatic capillaries run alongside with the blood capillaries, which allows exchange of fluid. So the excess fluid from the tissue and the blood is going to enter in the lymphatic capillaries. And from the lymphatic capillaries, they're going to go in the lymphatic ducts and be stored in the large lymphatic organs. Now, the lymphatic vessels, they are very similar to veins. They are very similar to veins in the sense that they are going to have valves. You know that large veins, not all veins obviously, only large veins have valves that are going to prevent backflow of the blood when pumping blood against gravity, right? So the lymphatic vessels are going to have valve system that are just going to play the same role to prevent a backflow. The lymphatic vessels are going to have capillaries, vessels, and ducts. The ducts being the largest pipes, right? Their function is to return the tissue fluid, right, which is going to be made up of water, salts, cellular products, including proteins, right? And all of that is going to be returned to the bloodstream. Now remember that the blood is made up of formed elements and the plasma, right? The plasma, it's water, salts, and proteins, right? And notice that here we have water, solutes, and cell products. Cell products are what carbohydrate proteins. So basically, we are talking about the plasma that leaves the bloodstream, right, to go in the lymphatic vessels, all right? But when that plasma gets in the lymphatic vessel, we don't call it plasma anymore. We call it lymph. By the same token, the lymph can leave the lymphatic vessels to be dumped in blood vessels, and in that case, we don't call that lymph anymore. We call it plasma. All right? So, as I already mentioned, the larger vessels in the lymphatic vessels are going to be made up of valves, okay, that are going to be there to prevent backflow. There are two major types of organs in the lymphatic system. We have the primary organs and the secondary organs. The primary organs are in two kinds. We're going to have the red bone marrow that occurs at the extremity of long bones. This is where all blood cells originate. And the second primary organ, it's the thymus. The thymus, which is the organ that sits on top of the heart. Now, the red bone marrow, it's where blood cells originate. That red bone marrow is going to be the source for white blood cells or the leukocytes, right? So without red bone marrow, forget about production of leukocytes. That is the reason why the red bone marrow is a primary organ of the lymphatic system. Two, the thymus. The thymus is where blood cells, and very specifically T lymphocytes, are going to learn the difference between self and non-self. By learning the difference between self and non-self, they are going to tell what is antigen and what is not antigen. Now, you understand that if T cells are not able to learn to tell apart the difference between self and non-self in the thymus, this is a problem, because now T cells, not being able to tell apart what is self and what is not self, they are going to cause issues in our body. That's typically when we have an autoimmune disease. Now, I have to say that in young individual, you have a huge amount of red bone marrow and a rather large thymus organ in proportion to the body, obviously, right? So this, in comparison to elderly people, so as you grow old and as you pass the age of 30 and you enter your 40s, 50s, 60s, the amount of red bone marrow decreases, the size of the thymus decreases as well, which means that your capability to protect yourself against infection is going to decline as you get old. While when you are young, that these primary organs are to the best of their shape, and they're going to protect you like never before, right? Or never after, rather. Let's put things that way. So as you get old, your immunity declines because your primary organs of the lymphatic system are decreasing in size and in quality. That is a very important information to remember. The secondary organs are going to include the lymph nodes and the spleen. The lymph nodes are where the leukocytes are going to be stored, waiting for an infection to occur. The spleen is going to be a site of storage, but very importantly also a site where the blood is being cleaned from debris, cellular debris, and so many things that have nothing to do in the blood. Now, the red bone marrow is going to be where blood cells are produced, right? We're going to have more bone marrow in children than we do have in elderly people, where the amount of red bone marrow decreases as we age. Some white blood cells are going to mature in the red bone marrow. Actually, most of the white blood cells are going to mature in the red bone marrow except for T lymphocytes. Then we have the thymus. The thymus, it's basically going to be the largest in children, shrink as we age. Immature T cells are going to go in the thymus and learn the difference between self and non-self. After they've learned that job, about 95% of these T cells are going to stay in the thymus. The reason being that we don't want to release a large amount of T cells in circulation in the blood. They can go there and start causing issues. So we're only going to release these T cells only as needed. The secondary lymphatic organs, the lymph nodes, they are going to be filled with lymphocytes and, very importantly, B cells and T cells. Macrophages, they are going to be abundant, the lymph nodes, in the neck, the armpit, and the groin regions. I want you to remember that the spleen is going to be found in the abdominal cavity, right? Its job is going to store the lymphocytes in the pulp, which is the middle segment, right? And the white pulp is in the middle segment. And then we have the red pulp that is on the edges, where filtration of the blood is going to happen. All right. So now that we've said this, let's address the different layers of protection. There are 3 levels of protection. We have the primary line of defense, the secondary line of defense, and the third line of defense. Now, the 2 first line of defense are part of what we call the innate immunity. The innate immunity, it's basically built-in immunity. This is the immunity you are born with. It doesn't require any previous awareness. It's just a primary defense system, right? And that primary defense system is going to be made up of the first line of defense and the second line of defense. In the first line of defense, we're going to have physical barriers. So barriers to entry, that's part of the first line of defense, right? Physical barrier. But we're also going to have a chemical barrier as part of the first line of defense. We have oils on our skin, we have salt. We are going to address all of that. All right. So we have a protective barrier as first line of defense. And then, what if there is a breach in that first line of defense? When there is a breach in that first line of defense, then, for instance, when you have a breach right here, then you have some infectious particles finding themselves inside. When these foreign particles come inside, there is no previous awareness, right? So you need a general purpose protection system there. And this is where the second line of defense comes into play. That second line of defense is going to involve some white blood cells, including dendritic cells, other white blood cells, macrophages, neutrophils, natural killer cells. These are all cells that are involved in the second line of defense. But you also have non-cellular components of the second line of defense. These are some proteins, including the complement proteins and the interferons. These proteins are in circulation in the blood. So we have these proteins here. They are in circulation in the blood, and these proteins are there to make sure that the protection system in our body is active against foreign particles. Other chemicals that are very important in the second line of defense are the cytokines. The cytokines in our immune system are basically the molecules that act as a voice to spread the news that there are some foreign particles in our system and we need to fight them. So they are kind of like the voice of our immune system, voicing that alert, alert, there are some infectious particles that are in, we need to mobilize and to go against that particle, right? And this is when, see for instance here, so we have some white blood cells that are in circulation and some white blood cells that are leaving the blood capillary to squeeze out of the blood capillary to go in the tissues where the infection is happening, where the breach is located, right? And I did tell you that squeezing between cells to go in the site of infection is called diapedesis, right? That diapedesis is made possible because the cytokines are in circulation and are triggering a number of events that cause these monocytes to be able to squeeze out and eventually to differentiate in macrophages. And as macrophage, they are able now to engulf foreign particles. All right? So, I told you about the first line of defense and the second line of defense, right? As part of the innate immunity. Now, the first line of defense is going to be both physical and chemical. The second line of defense is going to be both chemical but also cellular. So we're going to have some phagocytic cells, including dendritic cells, macrophages, neutrophils, right? We're going to have some protective proteins, including the complement and the interferons, but also something I didn't mention in the previous slide, an inflammatory response. Actually, to go back here, we do have an inflammatory response as part of the second line of defense. That aspect is very important to remember. Okay, so let's move on. Now, the physical barriers are going to include the skin, right? We are going to have the tears, saliva, urine. They are all ways to fight infections, right? They're going to flush out microorganisms that are a problem to us. We have the mucous membranes. Think about your mouth. You have mucous membrane in your mouth, in your eyes, in your nose, right? To mention only these mucous membranes. You have mucous membranes in your respiratory tract, right? The digestive tract, right? So respiratory tract, digestive tract, reproductive tract, the urinary tract. They are all areas where you're going to have mucous membranes. And then, considering the first line of defense, microorganisms are also going to play a very important role in the first line of defense. How is that possible? We do have on us and in us some bacteria that we call resident bacteria or the normal flora. These resident bacteria are occupying a physical space that basically newcomers are not going to be able to occupy for the single reason that this normal resident are just occupying the spots, right? And also, the resident bacteria, they are going to make sure that the newcomers don't get access to food and that they don't get established, which means that some resident bacteria can produce some chemicals that are going to be deterrent for newcomers to come and establish. Therefore, you understand that the normal resident, the normal flora, or the resident bacteria on your skin, in your mouth, in your nose, in different parts of your body, they play a very important role in protecting you against infections. And then we have the chemical barriers. In the chemical barriers, we have all the secretions. You have the oil glands in your skin. Please review the integumentary system. The sebaceous glands, they produce oil. That oil, actually, it's a deterrent to the bacteria that are non-resident. In the saliva, the tears, and the sweat, you do have, in the sweat, you do have the salt, right? That is a problem for bacteria that want to come and multiply, okay? So that is going to be a barrier. And then you have lysozymes. The lysozymes are very abundant in the saliva, in the tears, and these lysozymes is going to be very effective at breaking down the cell wall of bacteria. Okay? And then we have some environmental condition in some of our organs, like an acidic environment in the stomach is going to destroy microorganisms that we ingest. The vagina is also an area that is acidic, where the multiplication of microorganisms is also going to be controlled because of the acidity, right? So that's the first line of defense. The second line of defense is going to include the phagocytic cells, including neutrophils and macrophages. Now, you also have dendritic cells, right? Both are going to be able to circulate through the body and go in tissues where they are going to provide protection. Remember about diapedesis, right? Where the cells are able to squeeze between epithelial cells in the capillary level, right? So that they can go in the tissues. And these cells are also very important for the inflammatory response. Now, there are four major aspects in the inflammatory response. Four hallmarks, symptoms that are characteristic of the inflammatory response. One, it's redness. Two, it's heat. Three, it's swelling. And four, it's pain. Now, the redness occurs because of vasodilation. With vasodilation, during inflammatory response, you have an expansion of the blood vessel. They get so close to the surface of the skin that your skin looks red. That's the redness. Now, that vasodilation also goes alongside with a high flow of blood. That high flow of blood is going to create heat. That's why you have that heat. The swelling is because of the vasodilation. And the pain is because of the stretch that occur during vasodilation, right? The swelling, the stretch is going to happen and pain is going to develop. So four features that are very characteristic to inflammatory response. During an inflammatory response, you have histamine that is released by mast cells. As histamine is released, this is going to lead to vasodilation. That vasodilation causes the capillaries to dilate. And as the capillaries dilate, they become more permeable, which is going to allow monocyte to squeeze between cells. And again, we call this diapedesis. Diapedesis. All right? So the phagocytic cells are going to be to squeeze between and find themselves at the location where the infection is happening and the inflammation can occur. We have an increased blood flow, and that increased blood flow is going to cause warmth or heat, right? And that heat is very important to inhibit the growth of the pathogens, right? So an inflammatory response is really a response to limit chances that the infectious particle is going to survive. Then also we have an increased blood flow during the inflammatory response. That increased blood flow is going to bring more cells in there. I told you about the natural killers. I told you about the neutrophils, the macrophages, the monocytes. The monocytes differentiate into macrophages, right? Okay. And what is also very important to remember is that the inflammatory response, it's relatively short-lived, right? It is short-lived, and it's actually very important that it be short-lived because it's only an emergency response, right? From that emergency response, a more specialized response is going to develop at a later time, right? But you need a reaction just right now to limit the spread of the infection. So here we do have a summary of the inflammatory response. So typically, you will have a breach that occurs in the skin. That injury is going to trigger a number of events, including the mast cells that will start releasing histamines, and that histamine that is released is going to trigger a number of actions, including vasodilation, right? Vasodilation. And that vasodilation is going to cause the monocytes to actually leave the bloodstream and end up in the tissues, right? The monocytes are going to differentiate into macrophages. The neutrophils are going to be activated. These phagocytic cells are going to engulf foreign particles, right? All these guys, the green guys, right? And also, as they engulf these foreign particles, they are going to release some chemicals that we call the cytokines. These cytokines are going to go in circulation to cause even more diapedesis, so more recruitment of white blood cells, so that we are going to have a number of white blood cells at the site here where inflammation is occurring, right? Eventually, we are going to see a blood clot that is going to form, right? We're going to see some platelets that are going to come in the area here, and then we're going to have the prothrombin activator that is going to come. The prothrombin activator is going to activate prothrombin in presence of calcium, and prothrombin will turn into thrombin. Thrombin in presence of calcium is going to activate fibrinogen, so that fibrinogen is going to form fibrins, right? Forming a network that is going to stop the bleeding, right? So this is basically a summary of the inflammatory response. Now, the second line of defense is going to involve a number of proteins, including the complement proteins. These complement proteins are very much present in the plasma. Okay, they are going to be involved in inflammatory response by attaching to mast cells, which is going to trigger the release of histamine and vasodilation and rush of the blood in the area. Warmth is going to develop, and so on. So this inflammation is going to attract phagocytic cells, right? The complements are also going to be essential in forming what we call membrane attack complex, or MAC. The membrane attack complex are going to attach on some bacteria, and viruses create holes in bacteria and viruses and cause these particles to burst, right? So that's the role of the complements. And now the interferons. The interferons are going to be particularly abundant when we are undergoing a viral infection. So viral infection is going to cause the production of a huge amount of interferon, right? And this interferon plays a very important role in controlling the infection of of viruses, Okay, so now here we have the membrane attack complex that come together and form holes on a bacterium here, causing that bacterium to leak some materials out, and eventually the bacterium end up bursting, as you can see here. Now, how about the third line of defense? The third line of defense, it's rather a specific immunity. That specific immunity is going to involve B cells and T cells. The third line of defense is going to help protect against pathogens that we are already know of, right? We already came in contact with these pathogens, and now we are developing very specific tools against these pathogens, right? This is when the B and T cells come into play. Now, these B and T cells are also called B and T lymphocytes. These lymphocytes are for specific immunity. Now, let's take a look at them one by one. So, the B cells. The B cells occur in two kinds. We have plasma cells and memory B cells. The plasma cell, their job is going to be to specifically produce antibodies. So the plasma B cells are kind of like factory of production of antibodies. That's what I want you to remember. While the memory B cells are these B cells that are going to be saved for a later protection, right? It's a memory. It goes somewhere in our body. It stays there silent until, for some reason, we get infected with an infectious particle we have a memory for, and then our memory cells are going to come out and produce antibodies against that very infectious particle we have a previous awareness of. All right, how about the T cells? The T cells actually play a very important role in regulating our immune response. They are regulating our immune system by way of helper T cells. These helper T cells are the T cells that regulate our immune response. But then you are going to have other T cells that are not so much involved in regulating the immunity more than they are actually involved in participating at killing some cells that are infected. That's the role of the cytotoxic T cells. The cytotoxic T cells are going to physically attack our cells that have sequestered foreign particles, including viruses, bacteria, and other antigens, and cancer cells, right? So we have cytotoxic T cells. Cyto means cell, toxic means toxic, right? So these cells are toxic for other cells. The memory T cells are just going to be just like memory B cells. They are going to be stored for future use.
Daddy, look what Corbin's mom gave us.
So now let's take a quick summary. A summary here, it's that, well, we are going to have our specific immunity that is going to be developed based on antigens we already encounter. The third line of defense is also called adaptive immunity or adaptive defense. That adaptive immunity is going to not either really. It's going to involve antibodies in antibody-mediated immunity, and cell-mediated immunity. Antibody-mediated immunity is going to involve B cells, while cell-mediated immunity is going to involve T cells. Now, let's take a look at these two types of adaptive defense one by one. In the first one, let's stop on the B cells. B cells have on their surface some markers that are called B cell receptors. B cell receptors. These B cell receptors are going to recognize free-floating antigens. This is an antigen that is floating in our body, so they're going to recognize that antigen, right? As they recognize that antigen, this B cell is going to be activated, and as the B cell is activated, there are a number of events that are going to lead to plasma cells. And remember that plasma cells are specialized in producing antibodies. But let me tell you that the antibodies that are produced by plasma cells are nothing but the B cell receptors that are released in circulation. In other words, a B cell that is not activated has on its surface B cell receptors. They are attached to the surface. Now, a plasma cell, it's a B cell that actually can release the B cell receptors, and at that point, we don't call them B cell receptors anymore, we call them antibodies. That's it. And now, a portion of these B cells are going to be stored as memory B cells. And these B cells are only going to come into play when we have another infection. Now, let's take a look at the cell-mediated immunity. In here, just like the B cell, the T cells have some receptors that are called T cell receptors. These T cell receptors are unable to recognize free-floating antigens. Let me repeat that. The T cell receptors cannot recognize free-floating antigens. However, the T cell receptors only recognize antigens that are presented by our immune cells that have captured the antigen and presenting that antigen. So this cell right here actually carries a fancy name. It's antigen, antigen presenting cell. Okay? Antigen presenting cell, or APC. Okay? So unless this antigen presenting cell is presenting an antigen right here, well, this T cell receptor is not going to recognize it. Now, we know that this cell is presenting an antigen, so this T cell is coming, and now this T cell is going to undergo what we call clonal expansion, but we're going to maybe dive into that later. In any case, now you are going to have that the T cells are going to be activated and ready to work as cytotoxic T cells, while others are going to be stored as memory T cells. All right? So you have the cytotoxic T cells in here. Now, how about the helper T cells? The helper T cells right here are the regulatory cells in our system. These regulatory cells are the one that trigger the B cells here to undergo differentiation to turn into plasma cells. The helper T cells are also the one that are activating the cytotoxic T cells so that they become cytotoxic. We found that this is not going to happen, right? The clonal expansion is not going to happen. Okay? Now, the helper T cells for a particular antigen are also going to develop into activated helper T cells and memory T cells. Okay? So really not only B cells and cytotoxic T cells are going to constitute memory T cells, helper T cells will also. Now, I was telling you about antigen presenting cells, right? That are presenting an antigen. So look at this right here, APC, right? That APC, it's an antigen presenting cell, okay, that is recognized by a helper T cell so that the helper T cell can be activated. All right. So, what is characteristic of B cells? They are responsible for antibody-mediated immunity against pathogens. They are produced in the red bone marrow and they mature in the red bone marrow. Produced and mature in the red bone marrow. They directly recognize free-floating antigens. Okay, they directly recognize antigen. And they undergo what we call clonal expansion. Okay, which means that only the B cells that recognize a particular antigen are going to undergo multiplication so that we have a clone of one particular type of B cells. And that clonal expansion is essential for the production of plasma cells and memory B cells. Okay? And plasma cells are going to produce antibodies. These antibodies are going to be sent out of the cell by secretion. Okay? So the B cells are going to have B cell receptors, as I already mentioned it, right? These receptors are very unique for a given antigen, very specific to antigen. So not all kind of B cells are going to undergo clonal expansion, no. They have to be very specific, right? So the cytokines are very important here. These cytokines are released by helper T cells, and these helper T cells are the one upon the release of cytokines that are going to trigger B cells to undergo clonal expansion, and upon that clonal expansion, that B cell is going to become plasma cell and then produce antibodies that are sent outside. And remember that antibodies that are secreted are nothing but the B cell receptors, right? Except that these B cell receptors are not attached to the membrane of B cells anymore. They are released. Very important to remember. Now, when you have an infection and because of your antibody-mediated immunity, you were able to overcome that infection, then what? Well, the then what is that after an infection is under control, the B cells that were at one time used to fight that infection are going to undergo apoptosis. That apoptosis or programmed cell death is going to cause the death of about 90% to 95% of all the B cells that were in circulation. so that you have a very small fraction of B cells that are left. That very small fraction is going to account for the memory B cells. Okay? So basically, this is what we show here. We show that you have some antigens that are free-floating. Okay? You have different kind of B cells here with different types of B cell receptors. Notice that only the B cell receptors that match the antigen are going to undergo clonal expansion. So this one will not undergo clonal expansion because the B cell receptor do not recognize the antigen. This one also will not undergo clonal expansion because they don't recognize the antigen. Only this one will undergo clonal expansion. And now, undergoing clonal expansion, notice this. So now we have the plasma cells that are releasing what was at one time here the receptors, right? Now these receptors are released. Now we don't talk about receptors anymore. We talk about antibodies and see how these antibodies are going to recognize antigens, right? In the process, you also have some cytokines that are released by helper T cells, and these cytokines are going to cause differentiation of B cells. We have some memory B cells here, okay? Now, all the cells that were producing antibodies, they have to stop producing antibodies. They have to undergo apoptosis so that there is no extra antibodies that are produced which can cause an autoimmune disease. So no, we don't want that. We're going to destroy all the cells that were specialized in producing large amount of antibodies, because at this point, no more infection. So we have everything under control. We have to destroy the extra guys. Now, let's focus on the antibodies. What are the antibodies? What is the structure? Antibodies are these proteins that are specifically targeted against antigens. These proteins are going to have a Y shape, right? That Y shape is going to, as we look at the detail, we're going to see that there is a trunk that constitutes the foot and two arms. So we have the foot and two arms, right? The trunk is going to be made up of an area that is the same, that is a constant region. That constant region determines the class of antibodies. Antibodies occur in 5 different classes. We're going to see that later. The arms of the Y are going to be variable. That is the reason why when one antibody can recognize a particular antigen, another antibody cannot recognize the same area of that antigen. No, that's not possible. Okay? Because it's a matter of 3D matching. Okay? So this is the structure of antibodies. So you are going to see that we have a constant region that is right here, and a variable region that is right here. For a given class of antibody, the constant region is going to be the same across the class. The variable region obviously is going to vary, right? Notice also that an antibody is made up of 4 chains of proteins. We're going to have 2 heavy chains. So this is one heavy chain, like this. That's one heavy chain. And another heavy chain here. And then we're going to have 2 light chains, 1 and 2 light chains. Okay? What are the different classes of antibodies? There are 5 of them. One is antibody G. Now, you see that rather than saying antibody G, you see IgG. Ig stands for immunoglobulin. Immunoglobulin. So immunoglobulin class G here. This immunoglobulin class G are the main antibodies that you're going to find in circulation in the body. They are also the antibodies that are able to cross the placenta barrier so that mom can give to the fetus a maternal protection and some level of what we call passive immunity when the baby is born. Okay? What is the function of the IgGs? The IgGs are going to bind on pathogens. They are going to activate the complement, and they are going to trigger phagocytosis by white blood cells. How about the next class? The next class, it's immunoglobulin class M, IgM. This IgM are antibodies that can be found in circulation. They are found in circulation. They are the largest in terms of size. They are the largest antibodies you are going to find in the body. They are the first antibodies to be produced during an infection. So say you have a flu. Whenever you have a flu, the first antibodies that are going to be produced against that flu is the IgM. The IgGs are going to come much later, right? So they are the first one that are produced, okay? They are also the first one that are produced in newborns, okay? What is their role, the IgM? They are going to be very instrumental in causing activation of the complement, okay? Next class, the IgAs. Immunoglobulin A, they are found in large amount in the body, okay? But specifically in terms of secretion, so you're going to find them in mucous membranes. You're going to find them in the saliva. You're going to find them in the milk, so that a baby who is breastfeeding is going to benefit of a certain protection from these IgAs, right? So the IgAs, they prevent pathogens from attaching on epithelia. That's why they are found in mucous membranes, right? They are going to be found in the digestive tract, in the respiratory tract, to name only those. Then we have IgGs. IgDs. IgDs are antibodies that are found on the surface of mature B cells. The presence of these IgDs on the surface of B cells means that the B cells are ready to fight off an infection. Okay? Then we have IgEs. IgEs are antibodies that are found as a receptor on mast cells, which means that these IgEs are going to be involved in allergic response, right? They are involved in allergic response and also in fighting off some parasitic worms. Okay, so that's the antibodies and the B cells. How about the characteristics of the T cells? T cells are going to be in charge of the cell-mediated immunity, right? Typically against virus-infected cells and cancer cells. These cells are going to be produced in the bone marrow. However, they don't mature there. They are going to mature in the thymus, right? These cells recognize a particular type of antigens that are presented by antigen presenting cells, right? So antigen must be presented by a cell that through a receptacle that is called MHC, major histocompatibility complex. Major histocompatibility complex. It's a long name. Major histocompatibility complex, MHC, right? That this MHC in humans are called HLA. Okay? Now, the cytotoxic cells, their job is to destroy non-self antigens that are presented by an antigen presenting cells. The helper T cells are going to secrete cytokines, and these cytokines are there to control immune response. Whether you are talking about cell-mediated immune response or antibody-mediated immune response, the helper T cells are going to contribute equally in regulating the immune response. Now, the T cells, they are going to have, just like B cells, they're going to have T cell receptors. Okay? These T cell receptors are going to recognize antigens that are presented by antigen presenting cells, APC. So whenever an antigen presenting cell is engulfing a foreign particle, they are going to present that foreign particle, fragments of it, right, as trophies. These trophies are presented on the receptacle MHC, major histocompatibility complex, right? In a human, that MHC is called HLA, again, human leukocyte antigen. And by presenting it like that, this whole complex is going to be recognized by a T cell, and the cytotoxic T cell now is going to cause that antigen presenting cell to undergo death. So the T cell will specifically recognize a combination between HLA protein and a piece of antigen. By recognizing that, with the assistance of the helper T cell, the cells will undergo now clonal expansion. Now, obviously, the T cell receptors are very specific of the antigen, right? Okay, so the helper T cells, the cytotoxic T cells undergo clonal expansion. Some are stored as memory, right? After the infection, most of the cells are going to undergo apoptosis, just like we saw in B cells, and about 5% is going to be saved as memory, right? So what we saw for B cells is also true here. So we are going to have an antigen presenting cell right here, in this case a macrophage. That macrophage engulfed a bacterium. That bacterium got broken down and fragment of a bacterium was presented here. So only T cells with these T cell receptors are going to go clonal expansion. This one won't, this one won't. Okay? Now, as this cell now undergo clonal expansion, because of the presence of the cytokines, right, we are going to have a differentiation of the cytotoxic T cells here, large amount of it, right? The extra is going to undergo apoptosis upon control of the infection, while a small fraction is going to form memory T cells. Now, the helper T cells, again, they are going to help by producing cytokines, right? And the cytotoxic T cells are going to come in contact with an infected cell. So this is an infected cell, right? And this is a cytotoxic T cell. So the two membranes are going to come closer together. So we're going to have some perforins. These perforins are going to form holes in the antigen presenting cell. And through these holes, we're going to have some enzymes that we call granzymes that are going to be pumped in the antigen presenting cell. And that antigen presenting cell is going to die because of the granzymes here, all right, causing the death of that cell. Now, immunity can be passive or active. Everything depends, right? But what is immunity? Immunity is the ability to fight against infections and against cancers. That immunity can be brought about naturally through an infection, for instance, or through a purposeful exposure, or artificially through medical intervention. There are two types of immunity, active and passive. Active, it's when your body is intentionally developing means to protect itself against infectious particles or foreign particles, while passive immunity, it's when the body acquires immunity without ever doing anything special, right? In the case of active immunity, you can develop active immunity after an infection, okay, or after immunization. So natural infection or immunization are going to trigger active immunity, right? The primary exposure is most of the time going to be short-lived, and it's also going to be slower to develop. While the secondary exposure, so the second time you get exposed to the infectious particle, this is faster, the response is going to be faster, and the response is also going to be stronger, right? This is what we learn as we get exposed to infectious particle and as we get immunized, right? So, active immunity is going to be usually long-lasting, can last months to years, or even your lifetime, okay? It's going to depend very much on B memory and T memory cells. Now here, you are shown that when an individual is exposed to an infectious particle, so the first exposure, purposeful exposure through vaccine, we're going to see a response that is going to be relatively short-lived, okay? And is going to be relatively low as well. However, as this response is wearing off, if you provide a second exposure, what we call a boost, right? That second exposure is going to provide, in a relatively short period of time, a very high response. So you can see how high this response is compared to the first one here. And that response is going to, it's going to take a while for that response to wear off. That's why it was recommended that during COVID-19, once you take your first shot, that you should receive a boost and even a second boost, right? To provide that tremendous protection. Passive immunity is going to be a type of immunity that really the body is not working for. It is given to the body, right? That immunity is short-lived. It can occur naturally through, for instance, a mom passing IgGs to the baby through the placenta barrier, or IgGs and IgAs obtained by the baby through breast milk. And then you have an artificial passive immunity, which is when you can inject antibodies to a patient, right, so that their immune system doesn't have to do the work, right? You provide them the antibodies that are going to directly go against the infectious particle. Now, let's conclude this chapter with allergies, tissue rejection, and immune system disorders. First, allergies. Allergies are hyperreaction to harmless substances. Okay? This hyperreaction or hypersensitivity is going to be to pollen, food, animal hair. Now, some allergies are going to cause immediate allergic response. That immediate allergy response is caused because of IgE antibodies, which are attached to the mast cells, and the basophils are actually responding to allergens by releasing large amount of histamine, and that release of histamines is going to cause an inflammatory response that is very specific, that is very typical of allergies, right? Now, you do have some allergies, some immediate allergic responses that are very life-threatening. These ones are called anaphylactic shock. This anaphylactic shock can lead to a drop in the blood pressure, and this is life-threatening, but not just life-threatening. Actually, I know people who have lost their lives because of anaphylactic shock. For instance, being allergic to nuts, okay, the food nut, right, can lead to anaphylactic shock, cause a blockage of airways, and individual is unable to breathe, and without early intervention, that individual can die for lack of oxygen, right? We do have delayed allergic response. This is, for instance, response to poison ivy. You get exposed to poison ivy, but you don't develop the response just the very minute you are exposed to poison ivy. It's many hours after you are exposed to that. Tissue rejection is basically when our body is recognizing something as non-self, right? Remember that the cytotoxic T cells are learning the difference between self and non-self in the thymus, right? And that the MHC, major histocompatibility complex, are the receptacles that are presenting the antigen. They are also the one that are recognized as non-self or self. So the cytotoxic T cells are going to be able to tell that, this organ doesn't belong here. Now, for the cytotoxic T cell to not say that, you need to provide to the patient immunosuppressive drugs, and also you have to also make sure that the organ transplanted in the recipient patient, the MHC as similar as possible. We talk about compatibility when the MHC proteins are roughly extremely similar. They can never be the same. They can never be the same. They are extremely similar. That extreme similarity is what is going to cause the body to tolerate that organ after organ transplant, obviously, provided that the individual is taking immunosuppressive drug, and this is going to last the remainder of their lives, right? And there are some attempts these days for a person who needs an organ transplant to isolate their own cells and grow organs out of their own cells. And this is in the hope that we are going to see less tissue rejection, right? But we still have a way to go to achieve that, though we've done a tremendous progress. And then, what are some disorders of the immune system? Some disorders of the immune system include autoimmune diseases. This is when cytotoxic T cells are going to attack our own body tissues as if they were foreign tissues. Besides cytotoxic T cells that can do this, antibodies can also do this. Okay? Some examples of autoimmune diseases are, for instance, multiple sclerosis, lupus, like at the beginning of this chapter, myasthenia gravis, and rheumatoid arthritis. Now, last but not least, we're going to have immunodeficiency diseases. Some examples of immunodeficiency diseases are AIDS and SCIDs, right? Immunodeficiency diseases, it's when basically our immune system becomes weak because our blood cells are not there to protect us. It can be helper T cells, it can be cytotoxic T cells, it can be B cells. In any case, our immune system is compromised. Okay? Because our immune system is compromised, we are unable to maintain good quality of protection. Therefore, this is an immunodeficiency disease. All right? So this is where we are going to stop this discussion. Thank you for your attention.